Krebs cycle. Now this Krebs cycle is also known as citric acid cycle and tricarboxylic acid cycle. Now this Krebs cycle was discovered by H.A. Krebs, who is a German-born British biochemist, and he won a Nobel Prize for this in 1953. Now this Krebs cycle occurs in mitochondrial matrix in case of eukaryotes, and it occurs in cytosol in case of prokaryotes. Now the net result of Krebs cycle is that for each acetyl coenzyme A molecule entering in the Krebs cycle, 2 carbon dioxides are produced. Okay, 2 carbon dioxide molecules are produced for each acetyl coenzyme A entering the Krebs cycle. Now the Krebs cycle begins with the condensation of oxaloacetate and acetyl group. Okay, oxaloacetate is made up of 4 carbons and this acetyl group of acetyl coenzyme A is made up of 2 carbons. Okay, so Krebs cycle starts. Okay, the step 1 of the Krebs cycle is all about the condensation of oxaloacetate and acetyl group. Now oxaloacetate reacts with acetyl coenzyme A and water, and this reaction produces citrate and coenzyme A. Okay, so in the first step of the Krebs cycle, oxaloacetate is reacting with acetyl coenzyme A and water, and due to this reaction there is formation of citrate and coenzyme A. Now this reaction, which is aldol condensation followed by hydrolysis, is catalyzed by citrate synthase. Okay, citrate synthase is causing the condensation of oxaloacetate with the acetyl group and formation of citrate. Okay, citrate synthase is that enzyme. Now this citrate don't have chiral center, but it has a potential to react asymmetrically with an enzyme that has an active site asymmetric. Okay, so if enzyme is having asymmetric active site, then with that enzyme the citrate is going to react asymmetrically even though it does not have any chiral center in it. Now such molecules like citrate are known as prochiral molecules. Okay, these prochiral molecules, even though they don't have the chiral center, they will react with an enzyme whose active site is asymmetric. Okay, so citrate is a prochiral molecule. Now this step 2 of the citric acid cycle is divided into A and B part, which we called step 2A and step 2B. Now in this what happens is there is an isomerization reaction. Okay, and in this isomerization reaction water is first removed and then it is added back, and this causes the movement of the hydroxyl group from one carbon atom to its neighbor. Okay, the hydroxyl group is moved from one carbon atom to its neighbor. Why? Because water is first removed and then added back. This is what happening in the isomerization reaction of the step 2 of the Krebs cycle. That is, an hydroxyl group is getting moved from one carbon atom to the neighbor. Now this isomerization reaction which we are talking about is catalyzed by this enzyme known as aconitase. Okay, aconitase is a non-heme iron protein, and the thing is that it is the target site. Okay, this aconitase is the target site for the toxic compound known as fluoroacetate, which is a pesticide. Okay, so fluoroacetate targets the activity of this aconitase, due to which isomerization reaction does not happen and due to which Krebs cycle don't occur. And that is how the fluoroacetate is showing its toxicity and it is acting as a pesticide. It is not due to this fluoroacetate, the Krebs cycle is not taking place. Now this fluoroacetate gets converted into fluorocitrate, and then this fluorocitrate inhibits aconitase, and that is how the citric acid cycle is blocked. Okay, so fluoroacetate shows its toxicity by getting converted into fluorocitrate, then inhibiting aconitase, and then at last blocking the citric acid cycle. Now in step 3, the isocitrate is oxidized and decarboxylated into alpha-ketoglutarate, which is also known as oxaloacetate. Okay, isocitrate is converted into alpha-ketoglutarate in step 3. In first of the 4 oxidation step of the Krebs cycle, the carbon carrying the hydroxyl group is converted into the carbonyl group. The immediate product is unstable, losing carbon dioxide but still bound with the enzyme. Now the oxidative decarboxylation of isocitrate is catalyzed by isocitrate dehydrogenase. Okay, this enzyme is causing the oxidative decarboxylation of isocitrate, which is resulting in the formation of the alpha-ketoglutarate. Okay, that is what is happening in the step 3 of the Krebs cycle. Now in step 4, second oxidative decarboxylation reaction takes place, which results in the formation of succinyl coenzyme A from the alpha-ketoglutarate. Okay, alpha-ketoglutarate is subjected to oxidative decarboxylation, which converts it into succinyl coenzyme A. So there is formation of succinyl coenzyme A in the step 4 of the Krebs cycle. Now alpha-ketoglutarate dehydrogenase, this is that enzyme which catalyzes this oxidative step, and because of this step there is production of NADH, carbon dioxide, and a high energy thioester bond in the coenzyme A. Okay, so in succinyl coenzyme A, this coenzyme A is having a high energy thioester bond. So this is how the first step of the Krebs cycles looks like. Okay, oxaloacetate combines with acetyl coenzyme A and it gives rise to citrate. Okay, there is condensation of oxaloacetate and acetyl coenzyme A, the acetyl group of acetyl coenzyme A, and that condensation produces your citrate. And this is how the 2A, that is second step of the Krebs cycle, the part A of the second step looks like. Okay, citrate is getting converted into cis-aconitate and there is a release of H2O, as you can see in this step. Now during the part B of the second step, the water molecule is added in this cis-aconitate and isocitrate is produced. Okay, and it causes a rearrangement of the atoms, as you can see. Okay, so in the second part of the second step, the water molecule is added and then this cis-aconitate, okay, this cis-aconitate gets converted into isocitrate. After that, this isocitrate is converted into alpha-ketoglutarate. Okay, so that's your third step. In third step, the isocitrate is converted into alpha ketoglutarate, and in this conversion, as you can see, carbon dioxide, NADH and H plus ion is getting formed. Okay, so during the formation of alpha ketoglutarate from the isocitrate, there is release of carbon dioxide, there is formation of NADH and H plus ion. Now in the fourth step, this alpha ketoglutarate is then converted into succinyl coenzyme A, and in this conversion there is release of another carbon dioxide molecule and there is formation of NADH. Okay, so your alpha ketoglutarate is converted into succinyl coenzyme A in the fourth step. Now in the fifth step of the Krebs cycle, this succinyl coenzyme A is converted into succinate, and then there is a release of these compounds in this conversion. Okay, so this is the fifth step of the Krebs cycle where the succinyl coenzyme A is getting converted into succinate. Now in the sixth step, this succinate is converted into fumarate, and in the process there is release of FADH2. Okay, FADH2 is getting released in the sixth step of the Krebs cycle, in which there is conversion of the succinate into fumarate. After that, in the seventh step, this fumarate is converted into malate and H2O is incorporated. One water molecule is incorporated in this conversion, and that is what the seventh step of the Krebs cycle is all about. The fumarate is converted into malate. And in the last step, that is the eighth step of the Krebs cycle, this malate is converted into oxaloacetate, and in the reaction NADH and H plus are formed. Okay, so let's have an overview of this Krebs cycle. So this is how the Krebs cycle looks like. It is beautiful. Yes, and so yeah, you start with oxaloacetate, and condensation of acetyl coenzyme A and oxaloacetate is happening. It is giving citrate, then citrate is giving cis-aconitate, then cis-aconitate is giving isocitrate, then isocitrate is giving alpha ketoglutarate, and then alpha ketoglutarate is forming succinyl coenzyme A, which then gives succinate, then succinates get converted into fumarate, then fumarate gets converted into malate, and at last malate again gives you the oxaloacetate with which the cycle began. Okay, so this is how the Krebs cycle looks like. Now step 5. Okay, let's have a look at each and every step in detail. We talked about till step 4, then we had an overview, and now we are going to discuss step 5, step 6, step 7 and step 8 in detail. Okay, so that is how it went. So in step 5 there is cleavage of the thioester bond of the succinyl coenzyme A, and this cleavage of the thioester bond is coupled with the phosphorylation of ADP and GDP, and as you know, if you have studied that the glycolysis, that this kind of formation of ATP, okay, this kind of phosphorylation of ADP which results in the formation of ATP is known as substrate level phosphorylation. So in step 5 you are having substrate level phosphorylation because of the cleavage of the thioester bond of the succinyl coenzyme A. Now this step is catalyzed, okay, the fifth step of the Krebs cycle is catalyzed by this enzyme known as succinyl coenzyme A synthase, whose other name is also succinate thiokinase. Okay, so succinyl coenzyme A synthase is causing the cleavage of the thioester bond of succinyl coenzyme A. Okay, succinyl coenzyme A synthase. So I made a small mistake there. I called it synthase where it is synthetase. Okay, so succinyl coenzyme A synthetase is catalyzing this step 5 of the Krebs cycle. Now this ATP and GTP are energetically equivalent. Okay, they have same amount of energy in them. Now this step, okay, that is the 5th step of the citric acid cycle, in which there is cleavage of the thioester bond of the succinyl coenzyme A, which results into the ATP generation. Okay, so this step is the only step of the citric acid cycle that yields a compound which has a high phosphoryl transfer potential, okay, through substrate level phosphorylation. Okay, so this step is the only step which results in the formation of a compound which has high phosphoryl transfer potential, and that compound is either ATP or GTP. Now animal cells has 2 isozymes of the succinyl coenzyme A synthase in their cells. Okay, one isozyme of succinyl coenzyme A is specific for ATP, and the other isozyme of succinyl coenzyme A synthase is specific for GTP. Okay, so animal cells have 2 isoenzymes of succinyl coenzyme A synthase. The one is specific for ATP and the other is specific for GTP. It is synthetase. Okay, succinyl coenzyme A synthetase. I don't know why I am making that mistake again and again, but it is succinyl coenzyme A synthetase. Now the GTP formed by the succinyl coenzyme A synthetase can donate its terminal phosphoryl group to ADP, and this can cause the ATP formation. Okay, and this is possible by a reversible reaction catalyzed by diphosphate kinase. Okay, so diphosphate kinase is that enzyme that converts the GTP into ATP. Okay, so succinyl coenzyme A synthetase will result in the formation of the GTP. Later, that GTP can donate its terminal phosphoryl group to ADP, which will result in the ATP formation. Okay, this can happen, and this is catalyzed by this enzyme known as diphosphate kinase. Now, in the cells of plants, bacteria, and some animal tissue, the ATP is formed directly from the substrate level phosphorylation. Okay, in this case, what is happening? Substrate level phosphorylation is resulting in the formation of the GTP, and that GTP is then converted into ATP. Okay, this is what is happening in this case. But in plants, bacteria, and in some animal tissue, there is no GTP. Okay, ATP is directly formed from the substrate level phosphorylation. Okay, that is how it happens in cells of plants, bacteria, and cells of some animal tissues as well. Now, in step 6, which is the third oxidation step in the Krebs cycle, FAD removes 2 hydrogen atoms from the succinate. Okay, there is formation of succinate in the step 5, right? Now, from that succinate, the 2 hydrogen atoms are removed by FAD in the step 6. Now, this reaction, okay, the removal of the 2 hydrogen atoms from the succinate, this reaction is catalyzed by this enzyme succinate dehydrogenase. Now, this succinate dehydrogenase is strongly inhibited by malonate. Okay, malonate strongly inhibits this succinate dehydrogenase enzyme, which removes 2 hydrogen atoms from the succinate. And this mevalonate is a structural analog of succinate. Okay, it means that mevalonate and succinate have a similar structure, and that is the reason why malonate is inhibiting succinate dehydrogenase from removing the 2 hydrogen atoms from the succinate. Okay, it is obvious, right? Succinate will not bind to this enzyme. Mevalonate binds, and that is the reason why there will be no action of succinate on the succinate. Okay, and this is the classical example of competitive inhibition. Okay, so this succinate dehydrogenase is strongly inhibited by mevalonate. Now in this step 7, there is addition of water to the fumarate, which places the hydroxyl group next to the carbonyl group. Okay, in the next step, there is addition of water to the fumarate, and this results in the placement of the hydroxyl group next to the carbonyl carbon. So that's all is happening in the step 7. Okay, now let's look at the step 8. Now the step 8, which is the last of the 4 oxidation step in the cycle. In the cycle there are 4 oxidation steps, right? So this step 8 is the last one. Now in this step, the carbon carrying the hydroxyl group is converted into carbonyl group, and this regenerates oxaloacetate, which is then used in the step 1 again. Okay, so step 8 is the last step. It is all about regenerating oxaloacetate from the fumarate. So what happens is that in fumarate there is in malate, from malate there is regeneration of the oxaloacetate. So in malate there is a carbon which is carrying the hydroxyl group. That carbon carrying hydroxyl group is converted into carbonyl group, and that is how the malate is converted into oxaloacetate, and that is how the regeneration of oxaloacetate takes place in the step 8, and this is a oxidation step. So NAD plus linked malate dehydrogenase, this is the enzyme that is catalyzing the oxidation of malate, which is resulting in the regeneration of oxaloacetate. So malate is oxidized into oxaloacetate by NAD plus linked malate dehydrogenase. This is what is happening in the last step, that is step 8 of the Krebs cycle. Okay, malate getting converted into oxaloacetate. Now when 2 pyruvate molecules are oxidized into 6 carbon dioxide molecules, okay, here it should be 6 CO2. So yeah, when 2 pyruvate molecules are oxidized into 6 carbon dioxide molecule via pyruvate dehydrogenase complex and citric acid cycle, and then there is transfer of electrons to oxygen via oxidative phosphorylation, this yields 25 ATPs. Okay, so 2 pyruvate molecules gives 25 ATPs. Okay, via pyruvate dehydrogenase complex, citric acid cycle, and oxidative phosphorylation. These 3 processes converts these 2 pyruvate molecules into 25 ATPs. That is what the point is. Now let's look at the fate of carbon in citric acid cycle. Now glucose is converted into 2 pyruvate, and then that 2 pyruvates are converted into 2-acetyl coenzyme A. Now this 2-acetyl coenzyme A molecules enter the Krebs cycles, and Krebs cycle converts this 2-acetyl coenzyme A molecules into 4 carbon dioxide, 2 ATP molecules, 6 NADH molecules, and 2 FADH2 molecules. Okay, this is the product, and these molecules are produced from 2-acetyl coenzyme A entering into the Krebs cycle. Now glycolysis and Krebs cycle, both of them are producing only 4 ATP molecules per glucose molecule by doing substrate level phosphorylation. Okay, glucose molecule is resulting in the formation of 4 ATP by glycolysis and Krebs cycle. Okay, now out of this 4 ATP, 2 ATPs were produced by glycolysis and the 2 ATPs were produced by the Krebs cycle. Now at this point, FADH2 and NADH account for the most of the energy that is extracted from glucose. Okay, FADH2 and NADH account for most of the energy that is extracted from glucose. Now electron escorts link glycolysis and Krebs cycles to a machinery known as oxidative phosphorylation, which uses energy of the electron transport for ATP synthesis. Okay, so like FADH2 and NADH, which are generated from glucose via glycolysis and Krebs cycle, are used in oxidative phosphorylation to generate ATP. Okay, you will understand this better when you will see the video of oxidative phosphorylation. So go and watch it.